The next part of the chapter cell biology is non-membrane cell organelle. Till now we have discussed membrane bound organelle, which are endoplasmic reticulum, mitochondria, chloroplast, lysosomes, etc. Now non-membrane cell organelle, here we are discussing the one that is centrosomes. Centrosomes are found in animal cells and in motile algae. It has two cylindrical structures called centrioles, which is surrounded by centrosphere. Centrioles are arranged at right angle and they are made up of microtubules. The main function of centrosomes, it helps in cell division, and it also contains some non-living cell inclusions, which are also called as ergastic substances in the cytosomes. Now one of the most important part of this chapter, it is the nature of bonds linking monomers in biomolecules. Now what are biomolecules? Biomolecules, like proteins, carbohydrates, nucleic acids and certain important biomolecules are collectively referred as macromolecules. Now these macromolecules consist of polymers whose monomers are linked through specific bonds. Like proteins are made up of amino acids. So amino acids are joined with each other through one special bond that is called as peptide bond. So we are discussing how different types of bonds are present in biomolecules. We will be discussing some of these bonds like hydrogen bonds, peptide bonds, glycosidic bonds, phosphodiester bonds. To understand the linking present in the monomers of the natural polymers. Now we'll see first peptide bond. Actually we have already discussed in detail about peptide bond in the chapter amino acids and protein. Now here you can see in this structure that protein consists of polypeptides, and these are the polymers of amino acids. These amino acids link through the peptide bond. Now here you can see there are two amino acids, amino acid 1 and amino acid 2. Each amino acid has two functional groups. One is amino group and another is carboxylic group. So this part is called as N terminus and this part is called as C terminus. Now here a peptide bond is formed. It is nothing but a amide bond and it is formed between the NH2 group of one amino acid and COOH group of adjacent amino acid with the loss of water molecule. A water molecule is eliminated here and there is a formation of bond between carboxyl group and amino group, that is a CONH linkage is formed, which is called as peptide bond, and here there is a removal of water molecule. The next is glycosidic bond. Now here we have already discussed glycosidic bond in the structure of maltose, lactose, sucrose in carbohydrates. Here in maltose, two glucose molecules are joined with each other by alpha 1,4 linkage, while in lactose one galactose molecule is attached with one glucose molecule by beta-1,4 linkage. So, glycosidic bond is present in disaccharides and polysaccharides. This bond is formed between two adjacent monosaccharides. Like peptide bond, elimination of water molecule is there, and as there is a elimination of water molecule, again it is a dehydration reaction. The detail of glycosidic bond is already discussed in chapter carbohydrate. Now the next linkage which is present is phosphodiester bond. Now here you can see it is the structure of DNA. In DNA double helix, the backbone of the strand consists of deoxyribose sugar, see here deoxyribose sugar, and phosphate group. Can you see? Now here this is one nucleotide which consists of sugar, nitrogenous base and phosphate group. It is attached with the another nucleotide which contains again nitrogenous base, sugar and phosphate group. So let me clean this. Here a ester group is formed by dehydration of an acid and alcohol group. In phosphodiester linkage, two acidic groups of phosphate radical form ester linkage with OH group of adjacent ribose molecule. Actually, this detail we will discuss in the structure of DNA. Only one thing you need to remember, that in DNA double helix, one nucleotide, this is one nucleotide, is attached with another nucleotide through a linkage. Here this is the linkage which is called as ester linkage. Here also this is the linkage, because this is a acid group and this is OH group, and all of you know when there is a combination of acid and alcohol, there is a formation of ester. Here in DNA, as two ester bonds are formed, it is called as, and the acid is phosphoric acid, so it is called as phosphodiester bond. Now the next is base pairing in DNA. The two strands, here there are the two strands of DNA. The two strands of DNA double helix are held together by hydrogen bonds between nitrogenous base of the opposite strand. This is one strand and this is another strand. Each strand has nitrogenous base, and these nitrogenous base, these are linked with each other through the hydrogen bonds. These hydrogen bonds are very specific. Adenine, you can see adenine can bond with thymine of the opposite strand, and here it forms two hydrogen bonds. While guanine, it can form bond with cytosine of other strand and it can form three hydrogen bonds. Two bonded nitrogenous base from opposite strands constitute a base pair. These form a base pair, and the length of DNA molecule is often denoted by the base pair. This pairing is very fixed, that adenine bonds to thymine or vice versa, and cytosine bond with guanine with 3 hydrogen bonds. So here we have finished about the chapter cell biology.